Assalamu alaikum. Today we are going to discuss immunofluorescence. Here is some objectives, learning objectives of immunofluorescence. What is immunofluorescence? Principle of immunofluorescence, types of immunofluorescence. We will discuss its sample preparation, and at the end we will discuss applications of immunofluorescence. So what is immunofluorescence? As this term suggested, it is about immunological system that is about immune complex, which is antibody-antigen complex. Suppose we have antigen, to put antibody on there, it forms the antigen-antibody complex. And from this complex, we get the signal of fluorescence. So what do? We target antigen antibody. Sometimes we target antigen. So depending upon this target, we get our desired results. By measuring the intensity of fluorescence, we can calculate the amount of antigen present there. Next, we will see absorbance, difference between absorbance, luminescence, and fluorescence. What is absorbance? Light absorbed by a specific wavelength. Luminescence, light emitted by a substance not resulting from heat is called luminescence. We can say that is cold light. Here is the difference between luminescence and incandescence. Incandescence is the light emitted by a substance as a result of heating. I hope you understand the terms luminescence and incandescence. Now, the third term is fluorescence. Fluorescence it includes two states, excited state and its emission state. So we can say that emission of light by a substance that has absorbed the light. Absorbed light is called what we call excited state. Absorbed light, in most cases, the emitted light has longer wavelength and the lower energy than the absorbed radiation. Here is some fluorescent-based bioassays. The first one is cell assay, protein immunological assays, biosensing, energy transfer based assay, and probing. So we can say that fluorescent-based bioassays have been applied in biotechnology and various fields in life science. For example, various fluorescently labeled antibodies have been used to detect specific organelles, cellular activities. Its cellular activities include its morphology, viability, and function. And cellular process, example, transportation, endocytosis, and receptor functions. Next, we will see principle of immunofluorescence. Albert Conz, in 1944, American pathologist, immunologist, and physician, he said antibody could be labeled with any fluorescent dye. So we can see that the visible and invisible radiation produced from certain substances is called fluorescence. Next is staining or visualization technique. We call it staining or visualization technique, but this is not too common staining technique. There are some specialities which make them very unique technique. The first unique feature is that instead of staining whole parts, only specific parts of the cell or molecule is stained or labeled. The molecule present on the surface of the particular cell or even the present inside the cell. Here you can see that the molecule is present outside the cell or inside the cell. So what we can achieve to stain specific parts instead of whole cell. One thing we can check whether the particular molecule is present inside the cell or not. So we can check its presence. We can check its exact location. We can say that what is the distribution of antigen throughout the cell. The second special feature is stains used. Fluorescent stain, fluorochrome are used. Monoclonal antibodies are used. As I told, its second special feature is the stain used, called fluorescent or fluorochrome. Fluorescent antibody or fluorochrome. These connect to monoclonal antibodies. So we can say that it is called fluorescent antibody. So fluorochrome and monoclonal antibody have two different functions. As I told, in this technique, only specific parts are stained. So these antibodies used for the particular purpose. These antibodies specifically binds to desired parts only. Suppose if we want to stain nucleus only, we used anti-nuclear antibody which only bind the nucleus. The fluorochrome, in another hand, emit colored light. Then it is excited another light of specific wavelength. So fluorescent emit colored light, we called it fluorescence. Here is the nucleus of the cell that is stained by a fluorochrome, that is DAPI. 4,6-diamino 2-phenyl indole. Here is some examples of fluorochrome. Fluorochrome and their excitation wavelength and their emission wavelength. There is DAPI, their excitation wavelength 359 nanometer and emission wavelength is 461 nanometer. As I told before, longer the wavelength, emission wavelength, lower will be the energy. So in this case, we can say that DAPI has longer emission wavelength and lower energy. While in another hand, in the excited state of DAPI, it has 359 nanometer, smaller wavelength and greater energy. Here is some fluorochrome EFB, EBFB, their excitation wavelength, emission wavelength, as well as CFP, excitation wavelength, emission wavelength. There is types of immunofluorescence. There are two types of immunofluorescence: direct immunofluorescence and indirect immunofluorescence. First, we will discuss direct immunofluorescence. This is the antigen coated well. Now we are going to add antigen and the antibody, which is the fluorescence. Fluorescent antibody is attached, that is attracted towards the antigen. Now what happens? Antigen antibody complex is formed. The fluorophore tags help in the production of fluorescence. Once you know the value of the intensity of the fluorescence, you can easily determine the concentration of the antigen. How we can determine the intensity of fluorescence? By the spectrophotometry method, we put the test tubes in it. It shows the value of the intensity of the fluorescence. Value is noted. Now, what we are going to do, we are going to plot a graph. So we give this, with the help of this graph, we can find the concentration of unknown antigen as well as known antigen. With the help of known antigen, standard curve graph is plotted. So, how to know the concentration of unknown antigen? Here you can see that standard graph, which is used for the known concentration of antigen. And for the unknown concentration of antigen, same antigen-antibody interaction is occurred with the help of the spectrophotometry method, we can calculate its value and the graph is plotted. Extrapolated graph is plotted for the unknown concentration of the antigen. Here you can see that unknown concentration of the antigen is plotted through extrapolated graph, which shows the exact concentration of antigen. Here is the application of direct immunofluorescence. E. coli, Nigeria, monocytogenes, Salmonella species, Listeria cytomogens, Haemophilus influenza type B, and rabies virus. Next, we will see indirect immunofluorescence. The primary antibody, which is not connected to the fluorochrome, it is bind to the target antigen. And after its binding, a secondary fluorescent antibody against the primary antibody, they are allowed to bind them. So, in this fluorescent antibody, attached to the target antigen through the primary antibody, what we call fluorescent antibody attached to the target antibody via primary antibody indirectly. So, we call it indirect immunofluorescence. It is more sensitive than direct immunofluorescence. What is the reason? Because in direct method, only one fluorescent antibody is bound to per target molecule. In case of indirect method, two or more antibodies bind to primary antibody, which is bound to the target. So in this case, light emitted more and target and image is clear. There is the application of indirect immunofluorescence, Treponema fluorescens sartalis. Sample preparation. Target tissue is embedded in paraffin bags. Very thin slices are prepared through an instrument, what we call microscopy. After thin selected tissue transferred to a clean glass slide. After transferring, the wax is removed with the help of de-waxing agent. Slide is ready for staining. First, we have to check the distribution of protein in microtubules by direct immunofluorescence. Add fluorescent anti-microtubule antibodies on thin slice of cell and incubate for some time. During incubation period, these antibodies, you can see here, these antibodies bind to microtubules into the cell like this. And many antibodies remain unbound, and these can be removed by washing slide with help of buffer. After this, slide is observed under fluorescent microscope. Fluorescence microscope will bombard this light of specific wavelength. This light of specific wavelength will excite the fluorochrome to emit colored light around the microtubules. Here you can see that colored light of microtubule is emitted. Here is the types of fluorescence microscopy: wide field fluorescence microscope, confocal fluorescence microscope, and total internal reflection fluorescence microscopy. Here at the end, we are going to discuss applications of immunofluorescence. It is used as a histochemical staining method for making particular protein visible using specific antibodies. For example, yeast, hormones, enzyme, and plasma protein, as well as tumor or foreign antigen. Immunoglobulins, immune complexes. It is used to score diagnostics and to provide additional diagnostic and prognostic information in autoimmune disease, transplantation, and glomerular disease. This is all about my topic. I hope you understand. Thank you so very much.